the biocompatibility and hemostatic ability of our beads. So in terms of aim 1, so first we started off by synthesizing varying hydrogel compositions. We changed the compositions by changing the concentration of alginate beads by using 4%, 5%, and 6%, while keeping the concentration of chitosan constant at 1%. The compositions of our bead are based on incorporating alginate, chitosan, and zeolite in a suspended solution in which they'll cross-link. We finally will dry these beads using a lyophilizer, which is a machine that freeze-dries beads based on retaining a substantial amount of porosity. Then in terms of characterization, we will first swell the beads, and we will try to choose the concentration of beads that has the optimal swelling. And swelling is very important because it physically acts as a barrier against blood loss, as well as concentrate the many coagulation factors involved in hemostasis. Our next characterization is the ninhydrin staining, and we will stain our beads with ninhydrin dye, which specifically binds to the amine groups on the chitosan, thus showing the chitosan and alginate distribution throughout the bead. So in this picture here, we have our hydrogel synthesis in terms of the charges. So if you look at this top circle with the negative charges, we first make a negative solution of zeolite and alginate. Then we drop that solution into a positively charged solution of chitosan and calcium. And after these solutions mix, they'll cross-link. Specifically, the cross-linkers are alginate and calcium. So as you see in the next picture, they're found throughout the entire bead. Zeolite will also be found throughout the entire bead. And the chitosan is around the outer surface of the bead. So this is kind of exactly how we made it. We loaded the alginate and zeolite solution into a syringe, shown in this video playing right here, and it drops into the positively charged chitosan solution, and then it cross-links into beads, which is in the second picture, and then we washed and then dried these beads with the lyophilizer. So then the next step would be swelling. We evaluated the swelling capacity of our beads by performing swelling studies in which we swelled the particles in PBS and then calculated their changes in mass over time. Here on this graph, we have the average percent change in the masses of our beads versus the time. We swelled the varying compositions of beads and measured them at 5, 10, 30, and 60-minute time points. As you can see, the 4% hydrogel particles show to have the highest swelling capacity within an hour, followed by the 5% and then the 6% beads. So if you look at this little blood clot at the side, we have two of the main obstacles we had to overcome, one of them being our drying protocol. We initially compared air drying the beads with a desiccator to freeze drying the beads with a lyophilizer, and we found that lyophilizing the beads was much more effective. We had to do several trials of each to come to this conclusion, and again, throughout this process, we had to standardize all of our drying protocols. Another issue we had was replicating literature results, and there were discrepancies between our initial results and results that we had read in literature, and to overcome this problem, we validated our own results by increasing the replication of our data through several trials. So here you can see our ninhydrin characterization. This white bead is our initial lyophilized bead, which we cut in half, and then we immersed it in ninhydrin solution, which again reacts with the primary amines on chitosan. The resulting bead, you can see that the bead is stained purple, a purplish red color, and that indicates the location of chitosan. The bead has an alginate core, but alginate is also found throughout the entire bead because, again, alginate is the cross-linker of the bead. So then in terms of our next aim, we will assess coagulation through a time-to-clot formation study. We will assess platelet activation and platelet aggregation through specific assays such as the ELISA assay, in which we will use antibodies to identify specific clotting factors and proteins of interest. Finally, we will do a biocompatibility study in which we will use different live and dead assays such as tryptophan blue exclusion. So we very briefly started into, we're really excited to start with blood, and we compared 3 different compositions. We compared a blood control, a zeolite powder alone with the blood, and our non-zeolite loaded beads. We recently tried this experiment and we faced a few challenges. We anticipate that we will overcome these challenges by modifying our current protocol and increasing the replication of our data. And now I'll pass it on to Anjali.
Our last and final aim is aim 3, which will look at the in vivo assessment of our beads using a sheep injury model. Sheep are a great model because they have similar vasculature size as well as coagulation and fibrinolytic mechanisms. The liver will serve as a site of injury since it is susceptible to damage from abdominal trauma, and such injuries have a mortality rate of 10 to 15%. We will apply our material to an incision made on the liver and validate hemostatic ability by measuring the time it takes a clot to form as well as the volume of blood loss. Finally, we will inspect the incision site using Carstairs method to observe the hemostatic blood composition as well as platelet interaction, H&E staining to observe tissue morphology, and IHC staining to observe specific markers for inflammatory responses. Status of our research. Currently, we have made significant progress on many AIM-1 protocols, including bead synthesis and loading, swelling studies, and hydrogen staining, as well as beginning AIM-2 with time-to-clot studies. We've also applied to a wide range of grants. HHMI grant was directly related to our biomedical research. St. Jude's was an extremely competitive medical research grant, and Baxter was an educational grant. We received the HHMI grant, however, did not receive St. Jude's, and later discovered we were not entirely eligible for the Baxter grant. We recommend all new teams look at a wide range of grants with guidance from their mentors. Projects can be expensive, therefore funding is very important. We've also had significant milestones as a team, including IRB approval and its successful proposal defense. IRB, International Review Board, is an ethics committee that evaluates all human-related testing. Since we will need to test our hydrogel using fresh human platelets, IRB approval is necessary. However, it is a very long and difficult process. We recommend all teams start this process earlier since it requires many steps and revisions. Our second biggest milestone as a team was we were able to successfully propose our project last year, where we received a lot of great feedback from our expert, Dr. Murrow, as well as the rest of the Gemstone community. Team composition and process. The two biggest points of advice we can give for new teams is to focus on communication and accountability. Communication has to be clear between your team and your mentors when you're doing lab work and through the documents you write. Although we are a fairly large team of 12, we are still able to communicate well with each other and our mentors. Some of the modes of communication we use are, we have a lab notebook for all lab work and results, and use Dropboxes for all data and documents. We also use Facebook and group text messages for quick and effective communication, and our mentors have an open door policy in which we can meet with them whenever we need to. Regarding accountability, we expect everyone to be accountable for their lab work, as well as other assignments such as literature review and protocols. We also use a peer critique system and point system to evaluate one's weekly contributions. We also have team reflections every semester to evaluate our current methods of communication and accountability. In conclusion, we have made significant progress on many AIM-1 procedures, including bead synthesis, swelling, characterization, and loading. Although we experienced some challenges with creating new protocols, replicating previous studies, coordinating lab schedules, and accountability, we were able to overcome these challenges using better communication, breaking into subgroups, and creating a point system. We plan to continue with AIM-2 in vitro studies, to finalize AIM-1 procedures, and complete AIM-3